Hello friends, today we are going to learn principle and processes of biotechnology. That is the third part. See, in this diagram, what you can see, one is ori. Ori means origin. Origin represents the origin of replication. This whole is a diagram of E. coli cloning vector PVR322 showing restriction sites hind 3 eco R1 BAM H1 SAL1 and PVU second PST1 and CLA1 ORA and antibiotic resistant genes ampicillin and tetracycline rope codes for the proteins involved in the replication of the plasmid. Okay, so this is E. coli vector PVR322 in which this is ORI means origin of replication where origin of replication takes place. After that, this is ampicillin resistance gene in which PS1 which is restriction site is present, PVU restriction site is present. This is E. coli R1 restriction site containing sequence. This is CLA1 and Hind3 containing sequence. These two are uh, these two are antibiotic resistant genes in which tetracycline contains BAM-H1 that is restriction in endonucleoside. After that SAL1 restriction endonucleoside. And here is a rope which is replication of the plasmid which involved in replication and which contain PBU second site as a restriction site. So here we are going to discuss more about uh, E. coli cloning vector. Cloning vectors this so as their name suggested, cloning vector are responsible for cloning of gene and insertion into a desired host cell. So what it really contains. So here we know that the plasmids and bacteriophages have ability to replicate within bacterial cells dependent on control of chromosomal DNA. So on the basis of control of chromosomal DNA, these plasmids and bacteriophages has ability to replicate within bacterial cells. So suppose any bacterial cell is present, inside that bacterial cell, plasmid or bacteriophage can replicate independently of chromosome of that uh, microorganism. Bacteriophages because of their high number per cell have very high copy number of their genome within the bacterial cell. Copy number means they can produce number of copies. Bacteriophages because of high copy number per cell have the high copy numbers of their genome within bacterial cells. Some of the plasmids have only one or two copies per cell, whereas others have 15 to 100 copies per cell. Their number can go even higher. So some microorganisms contain like E. coli contains one or two copies of plasmids inside them. Plasmids are nothing but DNA fragments present inside plasma, which are independent of chromosomal DNA, which have uh, different replication strategies and different replication number, copy number than original plasmid DNA, and which do not get affected by the number of chromosomes either. So some microorganisms contain 1 to even 15 to 100 copies per cell. Even the number can go higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. Suppose we have a foreign DNA. Foreign DNA in that case, BT Bacillus thuringiensis contains Cry1AC and Cry2AB gene. These are responsible for bac- uh, resistant to uh, bollworm. These are get inserted into BT cotton with the help of Agrobacterium tumefaciens model and gene gun method, both. So in this way, the bacteriophage or plasmid DNA, if will insert by using restriction enzyme and restriction site inside an ampicillin or tetracycline resistance, we can insert a desired alien DNA. Here, alien DNA refers to the non DNA which is a foreign to the cell, which is not present inside the cell. So it can be any DNA piece of DNA that have desired characters. So, and its production capacity can be increased on the basis of copy number of the plasmid or bacteriophage. Vector used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. So vectors today used are engineered. Engineered means genetically engineered in such a way that they can help linking of foreign DNA and selection of recombinants from non-recombinants. The following are the features of features that are required to facilitate cloning of vector. Origin of replication or ORI. Here ORI stands for origin of replication. Replication is nothing but process of producing two or more copies of DNA from single copy of DNA. This is sequence where the replication starts and piece of DNA when linked to sequence can be made to replicate within the host cells. This sequence is also responsible for controlling copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. So higher the copy number, higher the foreign DNA can get replicated. On the basis of this principle, if we will select a replication ori of replication containing bacteriophage or plasmid, so it's higher the copy number, then higher it will produce the foreign DNA. Selectable marker. In addition to ORI, the vector requires selectable marker, which is the identifying and eliminating transformant in non-transformants and selectively permitting the growth of transformants. Here, non-transformants means the cells which do not contain foreign DNA, and transformants means which contains the foreign DNA or which got transformated. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, and kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry a resistance against any of these antibiotics. So normal E. coli does not contain any antibiotic resistant gene inside its cell. So how can we identify then? The transformation is nothing but the procedure by which we can transform we can insert the piece of DNA inside a bacterium through ampicillin, tetracycline, chloramphenicol, and kanamycin markers, which which do not contained by E. coli. So if these markers are present inside the cell, it means they contain the foreign DNA as well as foreign plasmid, that is PBR 322. Cloning sites. So what are cloning sites? These are the specific sites where cloning can happen. So we will study it directly here. In order to link alien DNA again referred to the foreign DNA, is the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Restriction enzymes means we can cut the desired DNA into two parts by using specifically tandem repeating sites. Presence of more than one recognition sites within the cell will generate several fragments which will complicate the gene cloning. Ligation of the alien DNA carried out at a restriction site present in one of the two antibiotic resistant genes. So normally two or three antibiotic resistant genes are inserted inside the desired cloning vector. So what are the role of it then? For example, you can ligate foreign DNA to BAM H1 site. So if you can cut the antibiotic resistant gene into two and insert foreign DNA inside it, like tetracycline resistant gene in vector PBR322, the recombinant plasmids will lose tetracycline resistance. So as tetracycline resistance gene got broken down into two parts, therefore its ability to produce tetracycline get hampered. So because of that, resistance due to the insertion of foreign DNA but can be still selected out from non-recombinants by planting transformers on tetracycline containing medium. So if we grow the cells into the tetracycline medium, then uh, tetracycline gene, as it got resistance got broken down, therefore it does not grow on tetracycline containing medium. That is that is recombinants. The recombinants growing on ampicillin containing medium are transferred onto a bacterium containing tetracycline medium containing tetracycline. So if transformants growing on ampicillin containing medium, ampicillin resistant genes are present inside the transformants, therefore it can give resistance to ampicillin containing medium. So if you will grow ampicillin containing medium, the transformants, and we can replicate same colonies inside tetracycline resistant medium, then ampicillin resistant medium does not replicate on tetracycline resistant tetracycline containing medium, because of which we can select the recombinants. That is, the recombinants will grow ampicillin containing medium but do not contain tetracycline. But the non-recombinants will grow on the medium containing both antibiotics. In this case, one antibiotic resistant gene helps in selecting transformants, whereas other antibiotic resistant gene gets inactivated due to insertion of alien DNA, alien against foreign DNA. Inactivation due to insertion. This is a phenomenon by which foreign DNA can get inserted into a tetracycline or any antibiotic containing resistant containing uh, gene and get inactivated. So it is called as inactivation due to insertion and help in selection of recombinants. Selection of recombinants due to inactivation of antibiotic resist and is cumbersome procedure procedure because it requires simultaneous planting of two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants. Chromogenic substrate. Okay, in these recombinants, DNA insertion within the coding sequence of enzyme beta galactosidase, the results this results into inactivation of gene on for the synthesis of enzyme, which refer to the insertional inactivation. Again, insertional inactivation is nothing but inactivation due to insertion of foreign DNA. The presence of chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. The presence of chromogenic substrate, chromogenic means color containing substrate, gives blue colored colonies if the plasmid in the bacteria does not have insert. Presence of insert result into insertional inactivation of beta galactosidase gene and colonies do not produce any color. This the identifies as recombinant colonies. See, beta galactosidase is a chromogenic substrate consuming enzyme. So if we will insert foreign DNA inside beta galactosidase, then the transformants will not produce any color, while non-transformants can produce blue color. So on the basis of this difference, we can select the transformants. So fourth is nothing but vectors of cloning genes in plant and animals. For example, so what are the different vectors that are used for cloning genes in plants and animals? So obviously, E. coli is used PBR 322 is used for production of transformers E. coli cells. Okay, so like that, plants and animals. So what are the different vectors that are used for cloning of plants and animals? For example, Agrobacterium tumefaciens. So agrobacterium means which is used for plant cells, tumefaciens. A pathogen for several dicot plants is able to deliver a piece of DNA known as tDNA, that is transform tDNA, to transform normal plant cells into a tumor cells and direct these tumor cells to produce chemicals required by the pathogen. So Agrobacterium tumefaciens is used for transformation of several dicot plants. So it produces a tumor and tumor inside the stem, otherwise different tissues of plants, and this produces the required chemical uh, by pathogen. Similarly, retroviruses means ancient viruses that contain RNA as their uh, genetic material. Okay. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. So retroviruses, what can they do? If they will infect us, then they will produce normal cells to become cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. So, to produce different cloning vectors or different chemicals required substances, substrates inside human cells, uh, retroviruses are used, but they are under some guidelines. Tumor inducing plasmid, that is TI plasmid of Agrobacterium tumefaciens, has now been modified into a cloning vector, which is no more pathogenic to the plants but is still able to use mechanically to deliver genes of our interest into the variety of plants. If you if you will use Agrobacterium tumefaciens to infect the plant, then it can produce tumor cells. But TI plasmid is nothing but tumor inducing plasmid, which is present in Agrobacterium tumefaciens. So if you will infect the plant cells with tumor inducing plasmid, that is TI plasmid, then it does not produce any tumor cell, but it can produce the desired chemical substance that are that is of interest to us. Similarly, retroviruses have been also disarmed and now used for delivering desirable genes into animal cells. So inside the genetic therapy or gene therapy, retroviruses has been used by disarming them and delivering the genes required for the production of desired chemical components inside the animal cells. So once the DNA fragment has been ligated into a suitable vector, it is transferred into bacterial, plant or animal host, where it multiplies. So this was all about the cloning vectors. So thanks guys for watching this video. Subscribe to my channel, uh, Life Science and Life Science for you, and press the bell icon to get new video notifications. Okay, thanks.